So here's a complementary base fitting, which is the basis for recognition. The transfer RNA, if we look at the molecule itself, here's a cartoon, and here's sort of a fleshed out structure. It has this odd shape, and on one side is the anticodon, where the recognition takes place, and at the other end it carries the amino acid. And this is called CCAN because this is the basis, the three bases that the RNA ends in. So it's also called the acceptor RNA. So here is just another presentation of this cognate codon anticodon attach, which is only possible if the two structures are presented to each other in a very specific geometric way. So what actually happens is that if there's an mRNA encountered in the cell, then it doesn't just encounter a single ribosome, but an entire series of ribosomes. One speaks of polyribosome structure. So the same mRNA can be translated by multiple ribosomes, and so you find a series of ribosomes sitting there, and here the mRNA has already gone through a substantial amount of time, and a larger part of the polypeptide has already been produced. And here the mRNA has just arrived in a very small one, a small structure. So the same mRNA can be translated multiple times by an arrangement of ribosomes called polyribosome. So several polypeptides are made, which all fold into the same protein. Now we come to visualization, which has been my job. First of all, 1 nanometer, which is sort of a unit here that we refer to, is 1 billionth of a meter. The human hair is 80,000 nanometers. The diameter of DNA is 2.5 nanometers. The ribosome has a diameter of 25 nanometers. The ribosome is essentially 1/4000th diameter of a strand of human hair. Right here is the human hair, and this dot is the ribosome. Okay, so that gives you an idea of the scale. So the visualization that we have to attempt is of this little dot here. So with the light microscope, this doesn't work because the ribosome is on the scale of the shortest wavelength of light, which is exactly 25 nanometers. In the light microscope, the ribosome would only show up as a tiny blur. By X-ray crystallography, this requires crystals, which means that the molecules have to be arranged in exact order. But crystals of ribosomes in many interesting functional states could not be obtained. So there are only a few structures of ribosomes that have been obtained, and they are not in particularly interesting functional states. And so the really powerful method is cryo-electron microscopy. It can be done by imaging single molecules frozen in ice and composing a three-dimensional image from multiple projections. So the idea is the following. The molecule, such as the ribosome, is arranged in a thin layer of ice. When I say thin, it is only 100 nanometers or 1,000 angstroms thick. And the molecules are arranged in random orientations, and then we see projections on the film or on the CCD camera. These are very noisy projections. In order to get the molecules into this layer of ice, we use so-called plunge freezer. This is a very simple device which can be hand-built, but there are, of course, very elaborate commercial versions available which can be computer operated and so forth. But the basic idea is that we have an electron microscope grid at the end, which is held at the end of tweezers, and one puts a single droplet of a solution that contain the molecules onto the grid, and then one has to plot the excess away with a piece of blotting paper, so that a very thin layer of this liquid remains. And then this plunger plunges the grid down into a liquid ethane, which is on the temperature of liquid nitrogen. And from there, the grid is being transferred into the electron microscope and being kept on the temperature all the time. So how do we do three-dimensional imaging? Well, three-dimensional imaging can only be done by putting a three-dimensional image together from two-dimensional images, from so-called projections. And so there are mathematical theorems that show that if one has a sufficient number of projections from different directions, then one can reconstruct the object in three dimensions. This is the microscope that we have in Albany. It's a so-called Polara from FEI, and it's a 300 kilowatt machine, which actually it can use helium as a coolant, but we normally operate it with liquid nitrogen. So this is a very powerful machine that we're going to transfer to Columbia. I recently moved from Albany to Columbia University in April, and we will have the microscope here soon installed. So this is the kind of data that we get from three-dimensional, from the imaging. You see that each ribosome appears as sort of a flaky thing, which you cannot really describe very well because it is so completely superposed with noise. Now, the advantage of cryo-electron microscopy over X-ray crystallography is that molecules such as ribosomes can be visualized in the natural environment as they interact with their substrate. What do I mean with substrate? Well, tRNA, mRNA, and then molecules that have not talked about, which are factors such as elongation factor G and elongation factor TU that are helper molecules which help the entire process along. So advantage is that no crystals are needed. But the three-dimensional image is reconstructed from a noisy image of many different molecules acting independently. So all molecules must be trapped in the same functional state. So we have to assure that all molecules whose images we put together are exactly in lockstep. They have to be trapped in exactly the same state. The disadvantage of the technique of cryo-electron microscopy is that it's difficult to get to atomic resolution. And that's clear because we need to perform so many operations. We need to align these noisy molecules and have to ensure that they are all in exactly the same configurational state. So there are so many glitches in these processes that it has not been possible to get to atomic resolution.